Hello, my name is Benjamin Joel Callaway, and this is my partner.
Theodore Crescenzi.
And this is our presentation on screening to potentially find antibiotics derived from bacteria isolated from soil samples. The research used in this presentation was derived from the research we've done in our microbiology lab course. I'll leave the introduction to Theo as we start.
Thank you. The antibiotic-resistance crisis is a worldwide issue that will only continue to grow if efforts are not undertaken to find a solution. Antibiotics have greatly improved our lives ever since their conception. However, the pharmaceutical industry has hit a wall. There are very few new antibiotics being discovered. Thus, business is no longer a profitable endeavor to pursue. The overuse of antibiotics has run rampant in our population for decades, ever since the first antibiotics hit the shelves. This overuse is not just in humans either. Even livestock are affected by this crisis. Roughly 80% of all antibiotics sold in the US find their way into animals. As a result, the ever-adapting microorganisms have continuously built up resistances, rendering past antibiotics useless to help the sick. And with very few new antibiotics being discovered, this is becoming a true crisis. One initiative has students, during classroom hours, go out, collect, purify, and test screen for antibiotics from bacteria taken from nearby soil samples to both familiarize themselves with microbiology techniques and contribute towards this worldwide endeavor. The name of this initiative is the Tiny Earth Project, and from soil discovered in this experiment, it is hypothesized that isolated bacteria will contain anaerobic antibiotic producers due to the poorly drained and wet soil that we collected as a sample. My partner will now explain the various methods that we utilized for this endeavor.
The materials and methods we used for this project were broken up into multiple sections, and it's worth noting that all experimental procedures followed the Tiny Earth Research Guide manual. The soil sampling, serial dilution, spread plate, pick and patch, colony morphology, gram stain, screening, antibiotic production, selective and differential media, biochemical tests were all done as different steps in this experiment. Now that we covered a rundown of our materials and methods, now the important part are recognizing what our results are and what they mean in the future. First up, the serial dilution plates. The 3 dilution plates with the dilutions of 10 to the -4, -5, and -6 were created using that same soil sample. And then after growing them, we had to calculate the CFU per milliliters of the plates that showed the most appropriate number of colonies of single bacteria. The dilution plate for the 10 to the -4, we ended up calculating was 1.8 times 10 to 5. Next up were our streak plates. Our 12 single colonies that were isolated and purified from the 10 to 4 serial dilution plate of the soil sample were put into 6 different LB agar plates using the streak plate method. Purified colonies from each streak plate were then grown on a master plate. As shown in the picture below, you can see that we used 6 different dishes and we grew 2 isolates on each. Each streak plate showed growth within the streak, and streak plate number 4 was visibly showing the least of all other streak plates, but still enough to work with. Here is our picture of our master plate with each of the streak plate isolates grown inside of respective boxes, and the purified colonies from each were grown on it using the pick and patch method. Each pure colony saw sufficient growth, some showing even interesting colors. Isolates number 2, 7, and 9 slightly outgrew their designated area, but they didn't contaminate the other pure colonies. Examining our master plate and our streak plates for colony morphology, we wanted to see what the margin, color, elevation, texture, and shape were generally across the board for each of these organisms. 11 of the 12 isolates appeared to have a flat elevation that were growing, and the general margin around the organism was a split between either smooth or wavy. The dominant shape was by far circular. The gram stains that we performed, that we used on S. epidermidis, P. fluorescens, and the number 1 isolate that we picked from our master plate to determine if each was gram positive or gram negative, resulted in, showing on the image on the left to the right, left being S. epidermidis at 400 times, as are the other two, had a cockeye shape and was gram positive, showing a purplish coloration. The P. fluorescens, also at 400 times, was Staphylococcus and was gram negative. The same results were for the isolate organism number 1, being Staphylococcus and gram negative. Both, as I said, both the isolate number 1 sample and P.O. fluorens are gram negative organisms and have Staphylococcus shape. S. epidermidis was gram positive and had a cockeye shape. The spread patch antibiotic production is our next category. Antibiotic screenings of each sample were performed against escape safe pathogens, E. coli, S. epidermidis, and E. aerogenes to determine if any antibiotic producers were present. The first attempt at screening on a plate growing E. coli and a plate showing S. epidermidis showed all isolates testing negative for antibiotics. Upon a second attempt, the results were also testing negative for antibiotics. Except in this second attempt, we also screened against E. aerogenes. In the first attempt, we only did E. coli and S. epidermidis. The selective and differential media plate tests using isolate number 1, B. subtilis, and E. coli were grown on the eosin methyl blue agar plates, or EMB plates for short. These plates were selective towards gram-negative organisms and would differentiate between lactose-positive and lactose-negative organisms. Neither the isolate number 1 or B. subtilis grew on the EMB agar and are thus gram-positive organisms. Looking at the figure shown in the picture, you could see the red coloration of these EMB plates. On the left were our isolate number 1, and on the right were our E. coli and B. subtilis. In the photo, it may be hard to see, but E. coli showed growth with darker coloration on the EMB agar, indicating lactose-positive gram-negative organism. The biochemical tests we performed, being the catalase, oxidase, and gelatinase tests, were all in the efforts to see what their metabolisms were unique for each of these organisms. All isolates tested negative for the oxidase test, indicated by the 0 coloration each of them. Had they been positive, we would have noticed a purple coloration on each of their samples within the first minute. However, all isolates tested positive for the catalase test. Using hydrogen peroxide in the organisms, they produced the bubbles, which was an indication of a positive test. Here we go. Taking all of these results into account, I'll let Theo discuss the bigger picture and what these results actually mean for the Tiny Earth Project.
Thank you. The various experiments and tests performed on the bacteria isolated from a soil sample was done in hopes that we could potentially find new sources of antibiotics to assist in the fight against the antibiotic resistance crisis. The hypothesis, sadly, was not supported by the results, as all isolated bacterium in this experiment tested negative for any antibiotics that could be of use. The results only compound on the growing number of failed attempts, small or large, that have also looked to source new antibiotics for the world around us. The number of new antibiotics that we have discovered over time has dwindled a lot. From the late 1960s to the early 1980s, the pharmaceutical industry introduced many new antibiotics to solve the resistance problem. But after that, the antibiotic pipeline began to dry up, and fewer antibiotics have been introduced. The result of this experiment was hardly shocking. The soil collected was an extremely small sample compared to other efforts being taken to source new antibiotics. Additionally, the scope of the experiment was hindered by the amount of safe escape relatives that were available in the classroom laboratory setting. However, any effort, no matter the scale is valuable for research, wider undertakings, such as more samples or broader screenings, should be carried out in the future to even, if slightly, increase the odds of succeeding in sourcing new antibiotics.